Hello, my name is Mark Martin, and I'm an Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. I'm currently teaching a freshman writing seminar. It focuses on particular skills that students need for success as undergraduates: reading closely, writing clearly, group work, and verbal presentations. As a focus for this course, a hub around which it rotates perhaps, I decided to use symbioses and parasitism to keep student interests up. As part of this course, I thought it would be wonderful to have a virtual seminar from various individuals, and I've been very fortunate to have some wonderful people volunteer. Today, I would like to introduce science journalist Ed Yong, who writes the quite wonderful Not Exactly Rocket Science blog on the National Geographic website. Ed Yong has become a specialist in the idea of mind-controlling parasites, as well as how microbes interact with our everyday lives. As usual, I used several different blog posts by Ed, presented them to my students, asked them to come up with insightful questions based on their close reading, collated those questions, sent them to Ed, and he was kind enough to use Google Hangouts to join us one day. And I'd like to share that experience with you. We are very, very fortunate to have had Ed join us. We're all quite grateful. Please enjoy.
So, is there anything I'm missing here? You should be able to this one, because every time that I've done it, I just say accept and it takes me right there. So I want to make sure that's what everyone else's experience is, right? Okay. Here we go. That's just his picture.
Hello.
Thank you so much for joining us.
That's all right. Are we good? Can you hear me?
We are good. I am told I need to click on your face with the whole face. That's disturbing, I know. Madison, am I doing this right? Okay. So I want to take this moment to welcome my class to meet with Ed Yong, who's one of my very favorite science journalists. He has a background in science. I believe you work in cancer biology.
That's right, yeah.
That's right. And then he's one of the people that I really trust on the internet with regard to things involving science. He's also quite amusing on Twitter. But what I'd like to do is have everybody settle in, and I'm going to put him in the in your viewpoint so you can have a look at him. Ed, here's my class, and thanks for joining us. I'm working
down. Hello. Hey guys.
Take it away.
Okay. So hi everyone. So Mark's forwarded a lot of your questions over, and I'm going to try and work through as many of them as I can. Apologies in advance if we don't get to all of them. I've picked out a few that I thought looked quite interesting, so we'll just take it from there and see how we do. Obviously, if there are any questions, either from you or from Mark, just feel free to stop me and let me know.
I just wanted to point out to you, Ed, that they've read the all the material that I sent to you, some of which you recommended, a couple of more posts from you, and there are those science fiction stories which you can comment on or not if you choose. But they've seen both of your videos, and that's one of the reasons we all wanted to see you, is because you're such an engaging speaker.
Wait, which was the other video?
You did the Reality Club one, which is an expansion of the TED Talk.
Right, okay, yeah. Okay, cool. All right, so I think let's start with a question by Alice Altbaum. So this was about the coincidental killers piece that I wrote in Aeon. And Alice writes, you describe when Anthony van Leeuwenhoek first, I will never get this pronunciation of that correctly, that guy, first discovered the amazing microbial world of his own dental plaque. One thing I love about science is that discovery is an unending process, and what has been a scientific realization during your career relating to symbiosis or parasitism that has altered your view of the scientific process or changed the direction of your focus in research or writing. So I like this question a lot because it allows me to talk about a couple of things that that are very important to me. I'm a science writer And you'll see from reading the stuff I've written that I really love talking about science and making it fun and accessible to people, and I love it. It's a field that really interests me and gets me excited. But I'm also a journalist, which means that I always have to be slightly antagonistic with the areas that I cover. It is my job not only to explain and to make things sound cool, but also to point out where things aren't quite working, or to resist hype and exaggeration in the fields that I write about. And the answers to Alice's question speak to this tension between going, hey, wow, cool, science, and also the need to occasionally say, this is bullshit. So the first example I wanted to talk about has to do with a parasite called Leucocoridium. So I don't know if this was on the package that Mark sent to you, but it's a type of it's a type of flatworm, a fluke, that infects snails. Have any of you seen this? Do you know what I'm talking about? Okay, it doesn't matter if you haven't. The fluke infects snails, and it reproduces inside the snail, and it sends these brood sacs into the snail's antennae. Now, a brood sac is pretty much exactly what it sounds like. I'm not really going to sugarcoat it for you. So the worm basically creates this parcel full of little baby worms into the snail's eye, and it pulses in a really quite disturbing way. It looks kind of green and red and black and white, and it's all mottled and revolting. And the idea is that when the snail has these two gigantic pulsating sacs, it makes it incredibly conspicuous to birds, who are the final host for the parasite. So I remember hearing about this in David Attenborough's documentary Trials of Life many years ago, and he basically intimated that the parasite not only makes the snail more conspicuous, but changes its behavior. So you'll be familiar with this concept after watching the TED Talk and reading about parasites, this idea of behavioral manipulation. So in this case, the parasite apparently makes snails climb to brightly lit conspicuous leaves and stay there while these flashing egg-filled billboards in it that will want its eyes attract birds. Now, this has been a classic of the parasite manipulation literature for like almost 2 centuries. It was first described in 1835, and it's in all the textbooks. And I wrote about a study last year which really surprised me, because it revealed, it just said that there's no data to back up this idea that the parasite is manipulating its host. We know that the worm creates these brightly pulsating sacs, but the idea that it also changes the snail's behavior in a way that allows birds, that makes them more attractive to birds, has always been conjecture, and yet it's kind of established. It's almost fact. So this Polish group actually decided to test it. They went out into the field, they did experiments with the snails and with the parasites, and they confirmed that it is true. The infected snails sit higher up on the ground, they keep to better lit areas, they stay in more open spaces, they move around a lot more. In short, they are more effective bird bait. So it's all good, right? We have this long-standing idea about parasite behavior being confirmed. But what I want to get across to you is that this took effort, right? So it took people deliberately going out in the field and checking that this was true. And it could easily have gone the other way. There are loads of myths about animal behavior that have been debunked recently that follow the same pattern of a long-standing piece of a long-standing fact, in air quotes, that just goes down through books and natural history documentaries for decades and decades and decades, with no one checking it, and finally getting debunked. So the reason I think this is important is that these areas that I want to talk to you about today, parasitism, symbiosis, they really capture the imagination. And it's sometimes easy to think, I kind of know what's going on. Like, it makes sense that parasites should be manipulating the host in this way. So if you look at a weird piece of animal behavior, it's very easy to create scenarios in which a parasite is manipulating its host. But that's always something that we need to test and that we need to see evidence for. And I'm going to go back to this theme as in answers to later questions. But it's really important to me as a science writer, as a journalist, to look at what the evidence tells us, look at what studies are actually saying to us. And if we are going to speculate, if we're going to go beyond that, to be very clear about where that boundary lies. So my second answer to this question is slightly different. My experiences on social media, and by that I mean both my blog and my experiences on Twitter, have been really instructive in this regard, because it's allowed me to be in touch with a large number of people in the field, like scientists like Mark and like other microbiologists, other microbiome scientists. And it's fascinating to hear people discuss their own field, because you get a very different sense of it than what you would do if you just read papers or read books or listened to TED Talks. You get to understand which discoveries are interesting, which ones are sound and rigorous, and which ones you should maybe be a bit skeptical about. You get to understand what the controversies in the field are, and we can talk about some of that stuff today. But it's a fascinating insight into what scientists on the forefront of this bit of knowledge are actually thinking, and how they're reacting to this sort of developing area of science around them. Okay, so from that broad kind of ramble, let's go into something a little bit more specific. So Daniel Swan asked a question. Hello. Asked about Wolbachia and how it turns males into females. So I will explain that, but I like this question because Wolbachia is pretty much my favorite bacterium. If you write about microbiology a lot, you do eventually get a favorite bacterium. There is no shame in this. You will probably find yours as well. But mine is Wolbachia because it's incredibly widely spread. So I think I probably mentioned this in the post that Mark gave you to read, but estimates vary a lot. Some people suggest that it's in 2/3 of arthropod species, which is immense, right? So this is a group that's the most diverse group of animals on the planet, and here's a bacterium that is in the vast majority of them. It has been described as the greatest pandemic in the history of life, and I think that's a reasonable statement to make. And it's fascinating because of all these weird parasitic manipulations that it exacts. A lot of them are antagonistic towards males. So the bacterium kills males in some species. There's a famous case of a butterfly called the blue moon butterfly that lives in the Pacific. It's a gorgeous insect. It's black with these kind of iridescent blue dots on its wings in the males. And about I think a decade ago now, some British scientists discovered that there was a 100-to-1 ratio of females to males among this butterfly. So the males had practically been wiped out, and that was entirely because of Wolbachia. It's not entirely clear how this happens, but we know that the bacteria somehow kills the males in the embryonic stage. So the male embryos fail to hatch, and the female embryos not only then face less competition for resources, but actually do better because they get to then cannibalize the corpses of their dead brothers. It does other things too. Wolbachia sometimes feminizes males. So in wood lice, it does this. It interferes with glands that produce male hormones, so that as the insects are growing up, those that are genetically male develop as females anyway. In some species, it turns them asexual. So rather than having the usual male-female mating producing different offspring, you just get a line of females that reproduce by cloning themselves. And this was proven in very dramatic fashion by a man called Richard Stouthammer, who treated this line of asexual Wolbachia-carrying wasps with antibiotics, and lo and behold, suddenly they started producing males. So he managed to actually cure them of asexuality by destroying this one bacterium. Now, other microbes use some or more of these tricks, but only Wolbachia uses all of them. And I was just writing about the history of Wolbachia for the book that I'm writing about partnerships between animals and microbes. And one of the things I really love about it is that, hang on, I'm just going to bring this up to make sure that I get the details of it right. But it was discovered, I think, in the 1920s. Yeah, it was discovered in the 1920s by a collaboration between a pathologist called Simeon Wolbach, which is why it's called Wolbachia, and an entomologist called Marshall Hertig, and they were studying one type of mosquito, and they dissected it, and they found this weird bacterium in its body, and a few years later, Hertig formally named it Wolbachia in honor of his friend. And I like that story because Wolbach died a long time before the significance of Wolbachia became known, before anyone knew how widespread it was, before a lot of these reproductive tricks were being discovered. Wolbachia is now being added into certain types of mosquito in order to try and control the spread of dengue and other diseases. So it's big news. It's a big player, not just in modern medicine, but in the history of life on Earth. And this man whose name has been appended onto this bacterium had absolutely no idea that he had been grafted onto the greatest pandemic in the history of life, which I think is pretty cool. Okay, Emma Casey wrote a question about the post I wrote about the virus that liquefies caterpillars, and she asked about this term, the extended phenotype, which features in the post. She says, I was wondering if you could elaborate on what this idea really means and if it has any applications to humans. This is a pretty important concept when thinking about parasitism and about symbiosis. So the term was coined by Richard Dawkins, and it's about the extended effects that genes have on the not just on the creature that carries them, but also on the environment that that creature then shapes. So my genes affect the way my body is, right? That's my phenotype. So genes affect my hair color and my height and my eye color and my propensity to disease. That's my phenotype, the collection of physical traits that makes me me. But I also change the world around me. I write blog posts and I do talks and I build stuff. I mean, not me particularly, but other people build stuff like houses and furniture and cars and whatnot. And we change the world around us and we create things that are beyond the realm of our own bodies. And that's what the idea of the extended phenotype means. It's the fact that my genes, as well as shaping the way I look and this kind of lump of meat that is me, also influenced all these other things that I went on to do in my environment. So if you think about the rest of the animal kingdom, beaver genes not only build a beaver's body, but also create a dam. And a beaver's dam shapes entire ecosystems. The same applies to termite genes, creating not just termites, but termite mounds, and therefore shaping the landscapes of savannas or plains. And when we think about parasites, so in the case of the liquefied caterpillars, they're part of the extended phenotype of the virus, right? So the virus has its own genetic material, which builds the virus, but it also corrupts the caterpillar and turns the caterpillar into a sort of extension of the virus. And the same applies for a lot of the other parasites examples that we've seen on say, the TED Talk, like the hairworm that takes over the cricket and launches it into the water. I made the point in the talk that at some point you could consider these animals as extensions of their parasites, because they have very little control over their own fates and destinies. They are being manipulated by something else and the genes of that something else. So in this case, the caterpillar is the extended phenotype of the virus, the hairworm is the extended phenotype of the cricket. Does all of that make sense? Okay, cool.
You don't have to come forward if you want. You can come forward and tell him, or I can repeat it because I have a loud voice.
Yeah, I was just wondering where, like, in terms of human behavior, where I tend to look at like stocks and where it's just observation.
So she's asking Ed about human behaviors in the extended phenotype, which I think takes us to E.O. Wilson's sociobiology kind of tenuously, right?
Yeah, right. Okay, so was the question about whether whether parasites or our gut bacteria or whatever are affecting our own behavior? Is that what you were asking?
So she's wondering what the divide is, you know, and I guess this comes back to a theme in the course a lot. How much free will do we really have? And I know you and I share a thought that we're great fleshy marionettes, but right. And so I think that's a little bit where this is going. Is that fair enough?
Well, okay. So I think it's very easy to go too far with this. So in the case of like, say, the association between the emerald wasp and the cockroach, I made the point in the talk that once the wasp stings the cockroach, that's it. The cockroach is now kind of a dupe for the wasp, an extension of the wasp, and has no say in the rest of what it does. Now, that's obviously not the case when you think about the relationships between our microbiota and ourselves. You know, I could lose all my bacteria right now and would probably be fine. I wouldn't be in great shape, but I would still survive. I would still be me. And you know, it's clear that our microbes influence our lives. They influence the way we process our food and the way and our behavior, which I'll get onto in a second. But I definitely think it's too far to say that, you know, they're robbing us of free will, right? We don't, as I said in the start of this little thing, we need evidence for this sort of thing. And there have been a couple of interesting speculative papers recently that like hypothesis papers about whether microbes are actually influencing our behavior. You could easily think about ways in which they do, like you could think about gut bacteria releasing hormones that increase our appetite, so we would eat more, and more food goes into our body, which means more food for them, which benefits them. You could think about microbes promoting social behavior. So there's some evidence in a recent fruit fly study that was out this year of a microbial odors promoting aggregation. So, you know, microbes influencing the socializer behavior of animals. Now, does this happen to us? If it did, then you could envision that microbes would more easily pass from one host to another, so maybe it helps their transmission. All of these are plausible ideas, but, you know, remember what I said at the start. What we need is evidence that they actually happen. And it's very easy to come up with plausible ideas when thinking about the microbiome, because it's so complex and because it seems to touch on so many aspects of our lives. But what I want to know is, does it actually happen? And, you know, a few of you, so Emma was one, Darby Bosco was another, asked about how microbes affect our psychology and our personality. And there's definitely a very rich line of research on this. We know that there are communication lines going between our guts and our brains. There are nerves that run the full length. The vagus nerve is basically a phone line that connects bowels to brain. We know that gut tissue can release hormones that travel through the rest of the body and affect the brain. So hormones, the immune system can act as mediators, communications between what seem like quite distant parts of our body. And all the classic ways of manipulating microbes, so looking at germ-free mice, giving antibiotics, doing microbial transplants, giving probiotics. There's evidence from all of these techniques that changing the microbiome can alter behavior, at least in mice. So, you know, by behavior, I'm not talking about like the kind of very specific parasitic manipulations that that I talked about in TED, but more about things like changing propensity towards anxiety or ability to deal with stress. So there's a group in Cork that has done a lot of good research about this. They had a paper, for example, showing that one species of microbe, Lactobacillus, I think, changes the levels of neurotransmitters in the brains of mice. It reduces anxiety and depressive behaviors in those mice, improves their ability to deal with stress, and that depends on the vagus nerve, which I talked about earlier. But you know, that seems to be decent evidence that microbes in the gut have the ability to influence the way we behave, maybe even the way we think. But it's still hard to interpret those results, and the mechanisms are still unclear. And remember that we're still talking a lot about animal studies. So how does one interpret what a mouse in a lab is doing, and how does one relate that to human behavior? I think these are all open questions, and at the moment we have some very, very intriguing bits of data, but there are more questions than answers right now.
May I interject?
Yeah, go on, Mark.
I believe you've been aware, because it's been all over Twitter during a recent meeting, Sarkis Mazamian's group at Caltech have claims that there's a particular bacterium that can reduce the incidence of autism-like symptoms in mice, and I've been very concerned about the sensationalism involved with that. Now, Sarkis Mazamian's a fine scientist, but I think this takes us back to your original comment about proof and applicability.
Right, yeah.
And I have great concerns about, you know, I've often said that microbes are viewed as either devils or angels, and they're neither. They're indifferent, most of them.
Right, absolutely. Yeah, I'd agree with that. As you say, I've got a lot of respect for Sarkis and his work, and you know he does very fine science, and he's done a lot of work in this space. That study in particular, I think, it's very interesting. The results seem solid, but it's about the interpretation, as you say, like what does this actually mean? To what extent are the mouse models really representative of human behavior? And I've talked to Sarkis about this at the meeting we were just at, and you know he's very, I think he's very nuanced about how to interpret that, and you know he understands that it's the best we've got, right? So you do mouse models because they tell you something, but you have to be very clear about what they actually mean when translating across to humans. I think what's interesting about that study is that they find one particular microbe and one substance made by that microbe that does have an impact on mouse behavior. Now, the relevance to human autism, I think that's pretty much an open question, and certainly the autistic community has had its fair share of people, you know, providing misinformation. So I think we need to be very careful about how we discuss these results and talking about the fact that they do represent science at a pretty preliminary stage, a very interesting one, but but one that we need to take great care about in discussing. Did you want to pick up that thread anymore, or shall I just go on to the next thing?
I just wanted to interject that because of I know that there are lots of families with autistic kids that were all over that, and that took me back to what you said originally about proof and applicability.
Right,
So
right.
I didn't mean to throw off your pace.
No, no, no. I was going to talk about this gut-brain axis stuff anyway, so yeah, that's more than happy to do that. Jesse Winchell? Hello. Okay, so you asked, in your opinion, what is the most interesting symbiotic or parasitic relationship you've come across? So I was trying to think about this. I'm glad that I got this question with some notice, because usually when people ask me this, I completely freeze. Like, I've just been doing book research for the last 3 months, and I've got so many things running around in my head that that my usual reaction is to completely blank. But I've had some thought, and there's one system that I really love. It's the citrus mealybug. So this is a tiny little sap-sucking insect. It looks like it's small and it's white, and it looks like a little dandruff flake. In its cells, so a lot of insects have bacteria living actually in their cells. They're called endosymbionts. So aphids have them. The sap-sucking bugs in particular seem to have them a lot. And the citrus mealybug is one of them. So it's got an endosymbiont called Moronella, which lives in its cells. Now, the reason I think this is especially cool is that Moronella has another little bacterium called Tremblea living in it. So you've got like Russian nesting doll bacteria, right? Like it's symbiontception. It's a bacterium inside another bacterium inside an insect. Now it gets cooler. The reason, typically, a lot of the symbionts make vitamins with their hosts. So did you guys read the bedbug post? Is that where the Wolbachia question came from? No idea what I'm talking about.
No, not the bedbug post. That'd be
okay. No, forget I said that. Forget I said bedbugs. Right. So a lot of these endosymbionts provide nutrients for their hosts. So plant sap has a lot of sugars and good stuff in it, but it also lacks certain important nutrients like amino acids that the insects need to survive. So the bacteria help to supplement these otherwise deficient diets. They make amino acids that the insects can then use. So think of it as like a living dietary pill inside all of your cells. Now, Tremblea and Moronella both cooperate to make amino acids for the insect. Making amino acids is a complicated business. It involves a lot of different chemical reactions. So you take like a starter ingredient, you process it, you get another one, process that, so on and so forth, until you get the amino acids you want. So it's a long chain. And what's amazing to me is that all of these three part players, the insect and the two bacteria inside it, the chain winds between them. So one of them will do one bit of the reaction, pass the byproduct onto another member of like another part of the symbiosis. They will do something else to it and pass it over. So all of them are involved in this. It's like a conveyor belt that runs through a factory, that runs through like all three of those partners, the insect and the two bacteria. And there are other genes in the citrus mealybug genome which contributes to this process, and they are genes of bacterial origin. So if I say horizontal gene transfer, does that mean anything to you guys?
Oh, yeah.
Okay, great. So, you know, bacteria can swap genes. Sometimes bacteria can give genes to animals that they live in, and that seems to be the case here. But the catch is that the bacterial genes in the mealybug's genomes don't come from either of the two bacteria that currently live in it. They come from at least 3 different species of bacteria, none of which are currently found in the mealybug. So these seem to be symbionts that once existed in this insect, shoved genes into its genome, and have now disappeared. And they are also involved in this process, in this vitamin-making process, in regulating the symbiosis. So really, you've got this tiny insect, and it's a mashup of 6 different species, 3 of which aren't even there anymore. And that that to me shows just how nuts these systems can be, and how unexpected they can be. You know, you never think, like, people have cracked open umpteen insects and found bacteria living in their cells. You'd never have thought that if you looked inside the bacterium there'd be another one. And in fact, people did see the other one and thought, it's probably nothing. So yeah, I think the mealybug symbolizes to me how complex and how constantly surprising this field of research can be. Okay, Lizzie Han. Hello, hand off the corner of that guy's head. So Lizzie asks, is it possible to develop helpful bacteria in the lab that can be introduced into individuals that lack them due to, for example, C-section rather than vaginal births, or bottle feeding rather than breastfeeding, or a history of malnutrition? How actively are researchers pursuing this? Pretty damn actively, as Mark can tell you. Now, a lot of this is on my mind at the moment, because I'm writing a piece about this right now. Firstly, we don't have to develop helpful bacteria. We can use natural ones. You know, there are stories of sorry. I know, for example, that Maria Gloria Dominguez Bellows' group is doing an interesting trial of what are basically vaginal microbiome transplants. So they're taking C-section babies and taking swabs of vaginal bacteria from the mums and basically just swabbing the babies with them to see if those vaginal microbes then colonize the infants, and perhaps even what happens, like how it affects the development of their microbiome later on, or even their own health. So that sort of stuff is going on. You know, other that I visited a group in UC Davis which are doing a trial giving a bacterium called B. infantis to premature babies. So, do you guys know about the sugars in milk that nourish bacteria?
They've heard about prebiotics.
Right. Okay. Great. So milk, human breast milk, contains lots and lots of sugars that nourish bacteria in the baby's guts, and in particular, they seem to nourish this one subspecies called Bifidobacterium longum infantis, or just B. infantis for short. Now, if you have the idea is that B. infantis has benefits for the infant in several ways, and that the sugars also help to prevent harmful bacteria from growing in the baby's gut. Now, premature babies tend to have problems with their guts. They tend to not have the usual cohort of bacteria assembling in their guts. So this group is considering using B. infantis and some of these sugars in breast milk as kind of a starter package to see if you can trigger the assembly of a more typical cohort of bacteria in these babies, and to see whether it can help to prevent some of the health problems that they develop. Now, clearly, there's a lot of potential here, but I think it's important to sound a note of caution as well. We're in quite early stages of understanding how communities of microbes assemble and how they change over time, about properties like stability and resilience, and how different communities vary between disease and between health. And it is becoming, I think, increasingly clear that there isn't any such thing as just one standard healthy microbiome. That health varies depending on a lot of different contexts, on person, on maybe which part of the world they come from, maybe what diets they were eating, maybe their own background. So it's very difficult to think about if you want to shift a person's if you want to shift a person's microbiome, what exactly are you going to shift them to? How are you going to shift them effectively, and what are the consequences, if any, of doing that? So you'll probably have heard about fecal microbial transplants, FMTs, where people who have quite intractable gut infections of C. difficile get treated get basically implanted with the fecal matter and the associated microbes of a healthy donor. Now, the technique has had a lot of promise, but this is a very specific use for it. People with C. difficile infections, it's very clear that they are not doing so well, and what you really want to do is to not have them not have a C. difficile infection. Now, it's a very different situation when you have a person who is otherwise healthy but has gone through what one might think of as an event that disrupts their microbiome, like going through C-section delivery or being bottle-fed rather than breastfed or going through malnutrition. To what extent do those events warrant the same sort of treatment? I really don't think we can answer that at the moment. I think we need to be very cautious about proceeding. But to answer the question, there is definitely a lot of interest there, and you should see a lot of development in this space in the future. I remember talking to a woman called Jo Handelsman, who's the pioneer of this field, at the recent meeting, and she likes to say that the microbiome is the only organ in the body that you can replace without surgery. And that is absolutely true, and that means that there's a tremendous potential here for shifting it in ways that might improve our health. But it also means that it's incredibly complicated, and that we need to really understand what we're doing before sort of proceeding in a kind of gung-ho fashion. Does that make sense?
So Ed, we're pretty much out of time for today. They have other classes to go to.
Sure.
I want to draw everyone's attention to a book I certainly will assign to classes in the future, and I hope to be able to review, which is going to be coming out in 2015 by Ed Yong.
2016.
2016, sorry. Called Contain Multitudes. I love me some Walt Whitman, so that's great. I am, after all, a microbial supremacist. But I wanted everyone to take an opportunity to thank Ed Yong for his time today. And it was very, very useful, sir. Thank you so much, and I'll talk to you soon.
Thanks, guys.